Take three, guys. You have no idea what I've just been through. I don't know how to edit yet, so I apologize. I'm gonna go with this take no matter what, but I can't edit, so it might be bad. Okay, this is the Protein Synthesis Lab Helper. I'm going to give you guys a little bit of instructions to clarify how to do the DNA, the code lab, since we are not in class to do the DNA code lab. I've printed mine out. You do not need to print yours. You can, of course, but you can also just do it as a doc. You can see I've worked on it quite a bit because this is not the first take. First thing I want you to do, though, is I want you to read. I want to make sure that you have a good overview of the whole process of transcription and translation, the process of going from the code in DNA to finally building a protein. And so you're going to need to recognize and remember this. DNA goes to RNA, and RNA goes to a protein. And in fact, we could even put a little like sort of mini step in here. RNA goes to amino acids in order to build the protein. Once you've read this, you're going to practice down here at the bottom. All right. So DNA is made up of adenine and thymine and cytosine and guanine attached to a sugar phosphate backbone. But those bases we represent with the letters A's and T's and C's and G's, and you guys know how they pair: A's with T's, T's with A's, C's with G's, G's with C's, right? Straight line letters go together, curved line letters go together. It makes it pretty easy to follow along. But the genetic code is read 3 bases at a time, and 3 bases at a time is called a codon. So this first 3 bases in a row is a codon. The next 3 in a row is a codon. The next 3 is a codon. A codon is a genetic word. And codons code for different amino acids. And so that's how the DNA code allows your cells to put different amino acids in the right order to make the right shaped protein to do a job. So I'm going to see if I can lay this down here. I'm going to not make a big mess of the pieces that I had already pulled out. Let's see here. Okay, so you guys are going to need to practice transcribing your DNA sequence into RNA. So the coding rules are the same: C's with G's and G's with C's. T's pair with A's, but there's one exception. A's can't pair with T's because there are no T's in RNA. Instead of thymines in RNA, there is uracil instead, and it has to do with the stability of the single strand, but that's the only difference. Otherwise, all of the rules are the same. But I've made this little cheat sheet down here for you to help you with your transcription. So if these are the letters in your DNA sequence, when you are writing out your RNA sequence, you're going to do these matches instead. So our first example here, A, T, C, actually is U, A, G. A pairs with not a T but a U. T can pair with A just fine. And C and G are the same, so C pairs with G. And G pairs with C. And T pairs with A, no problem. C with G again. And A pairs with U. You just have to remember there are no T's in RNA. So you're going to do this whole sequence, practicing it, and separating each codon. So that's what I've got these slashes here for, is to separate every 3 letters. So AUG, that's a codon. CAG, that's a codon. GU, that's another codon. A would be with a U. T can go with an A with a U, and that's another codon. So you guys are going to practice that, separating all 3. If you can't get all 3 at the end, right, there's just no more codons for you to see. So again, you're going to read to start with. I'll give you a quick overview of the diagram here. So here we've got a cell, and inside of the cell is a nucleus, and the DNA is inside of the nucleus. The DNA stays always inside of the nucleus. But in order to build proteins out here in the cytoplasm, we need to first make a copy of the DNA code in the form of RNA. So that's the process of transcription, unzipping the DNA, making a single-sided copy, and then that RNA can leave the nucleus and go out into the cytoplasm. When it gets to the cytoplasm, it's going to bond to a ribosome, which is going to hold everything in place while the process of transcription or translation occurs. There is a second type of RNA called tRNA, transfer RNA, that carries amino acids. It's not labeled on your lab, but I wrote a little AA here to show that this little dot here is an amino acid. And the amino acid is carried by its own tRNA. Remember, there's 20 different kinds of amino acids, and each one is shaped a little bit different. So according to the code on the RNA, where the ribosome is holding on to it, only one type of tRNA can match that code at a time. And so as the ribosome slides along the RNA, reading each codon 3 bases at a time, it's going to bring in different tRNA molecules that will bond according to the code and deliver a different type of amino acid that builds slowly a long amino acid chain that folds into the correct protein. So in the lab, if we were in the classroom, you would go to the nucleus, which would be a table at the front of the room that I would label nucleus. I've got this lovely little document attached to this, the lab helper, which has pictures from when we did this last year. So here you can see I made a nucleus on the desk, and inside of the nucleus is the DNA. You guys are going to be operating actually right from the picture in this document. So if you were to straighten out this DNA and separate it, so you're only looking at one side, you guys are going to look at the sequence that's in green. Don't worry about the promoter sequence. Don't worry about any of the other colors on here. You're only going to be looking at the green section here. But, and I promise you it's easier to see in the document. I know it's really hard to see this right here. But along this green sequence are different bases. And I've actually, what I've done here, I've borrowed Ella's leash. She doesn't care, she's a dog. And I've actually written it a little bit bigger, just part of it here. So here we've got our DNA sequence, and we are going to separate and look at only one side of the DNA sequence. And then you guys are going to write in your lab, you guys are going to write what pairs with the DNA sequence. So you can see on the DNA, and you're going to be looking at the green strip, we've got bases T, A, C, T, T, G, T, A, G, T, C, T, all right, on and on and on. What you guys are going to be doing is writing in, just like we practiced on the front page, what this pairs with. So you're writing your mRNA sequence, where it says start here, looking at what the DNA is going to pair with. So here the first 3 letters, T, A, C, actually pairs up with A, U, G, right? Because T pairs with A, and A pairs with U, and C pairs with G, and you're going to do that for the whole sequence. Now, I don't care if you do 3 letters at a time and then mark that that's one codon and put a slash there, or if you do the whole thing and then go back through and do 1, 2, 3 slash, 1, 2, 3 slash, 1, 2, 3 slash. It doesn't matter to me, as long as you can see what codons this section of DNA codes for. So you're going to take the section of DNA, you're actually going to be looking at this copy in the document attached to the assignment, and you are going to write on your lab or type onto your lab what the corresponding mRNA sequence would be. Once you've done that, in class you would take your code and walk across the room to a separate desk labeled ribosome, but I don't have a separate desk labeled ribosome, and you guys can't pick and choose from the bases there. So what you're going to use instead is something called a universal codon chart. Now, this is the way most students would do a lab like this, and there's two styles of chart. There's a square style that's attached to the document that you guys have reference to in Google Classroom, or you can just Google universal codon chart and look for this style. I like this style. It's easier to use. It's got all of the amino acids on the outside, and you start in the center. So here's where you guys actually get to do the work. So if my first codon is AUG, I want to find out what that codes for. So I take a look in the center, and I start with A, and then I go to U, and I go to G, and I see that it says methionine. Now on the other chart, it has only the abbreviations. I'm going to write in the abbreviations just because it fits. If you want to write really tiny, you can write out methionine. But that's what you're going to be writing on the line in your lab for your amino acids. So my first amino acid is methionine. I just wrote met. My second code, AAC, I take a look starting in the center. A, C is asparagine. And so that's what I'll write for my second amino acid. And the abbreviation for that is actually ASN. I screwed it up on the last take. Doesn't matter. My third sequence, I actually haven't written in here. Let's see. It looks like it's TAG. So let's see. TAG would be AUC. And if I look on the codon chart AUC, I have isoleucine. The abbreviation is ILE. Now you're going to do the rest on your own, and you're going to fill in all of the amino acids that are coded for by the green strip of DNA. Now, you'll notice also on these codon charts that there are 3 codes for stop. UAA, UAG, and UGA. All 3 of those code for stop. So if you get the code for stop, you're going to write in stop, and then you're going to stop. So I'm going to show you, though, what happens at the ribosome by following the code. So met is our first, methionine. And then we've got some ASN. Now, I've made these just random shapes, but in reality, each amino acid is a little molecule that has its own shape, and they only go together a certain way. The next one, isoleucine, ILE. And then let's see here, we've got some arginine, ARG. And then some glycine, GLY. And the way I've cut these out, and if we were in class, you'd actually get to play with them as well. They only fit together to form a certain shape, and you can see if you translate wrong, and therefore, or if you transcribe wrong, and then that means you translate wrong, you'll have the wrong shape, and you might not even be able to build a protein. Let's see here. Then we get, that was from glycine to glutamate, and then aspartate, and then we've got some valine, and another isoleucine, and a histidine, and a stop. So here I have just built a sequence of amino acids coded for by the DNA, put together in a particular order that makes, based on the shape of the amino acids, makes a shape of a protein. And this shape is just right to do a specific job in a cell. Maybe it's the structure, maybe it builds a molecule or takes a molecule apart. But if the protein comes out the right shape, it is functional. It can do its job. I will help with the questions once you get through the doing of the lab. you're going to need to do the questions. I will help you guys with that during a hangout. The next hangout will be Monday at 11. If you have any questions between now and then, feel free to email me or send me a message on the Google Hangout app. Emily just showed me how to do that. And otherwise, I will see you guys on Monday. Bye.